treatment and wait to see if there's a recurrence. Osteosarcoma is also one of the cancers that is resistant to immunotherapy. So kind of immunotherapy has been a huge revolution in oncology over the last probably 10 years. But unfortunately, there's a few diseases like osteosarcoma that are resistant to this disease. And the reason that osteosarcoma is resistant to immunotherapy is in osteosarcomas, they are surrounded by these M2 or cancer promoting macrophages. And that actually makes up about 50% of the tumor volume for most cancers. And so it basically sits there and makes this very nice, cozy and happy environment for cancer cells where they can grow and proliferate and also hide from immunotherapy drugs that are so effective in other diseases. So that is really what we're targeting with our therapeutic approach. So our goals are to develop an effective therapy for preventing metastasis in pediatric osteosarcoma. We hope to do this by exploiting the tumor microenvironment. And we've also been able to specifically deliver chemotherapy directly to the cancer cell. So I'm going to go ahead and I'm going to show just a little bit more background. So one of the functions of macrophages, there's basically two categories of them. The M1s are the anti-cancer type. And so they actually seek out tumors, they home to the tumor and they secrete cytokines and kill those cancer cells off. And I was mentioning the M2 are the cancer supportive ones. They hover around the cancer and again, make that really nice and supportive environment where cancer cells can grow. And the interesting thing about these is that they can convert back and forth between these two different phenotypes. So here's our M1s or our anti-cancers, and here's our M2s on the other side in pink. And we call this repolarization. So basically they can go back and forth from pro-cancer to anti-cancer. And so our strategy is actually developed to convert those M2 macrophages that are hanging around by the tumor and convert them to the M1 or good or anti-cancer phenotype. So does somebody have a question? I'm sorry. And again, this is just kind of and the strategy we're using to do this is with our M1 nubs, and I'm going to explain a little bit more about those as well. So extracellular vesicles are things that are endogenously released by your cells. So as we're sitting here right now, our cells are releasing them. They function in cell-to-cell communication, and they're little kind of blobs, 150 nanomolars. And so what they can do is they can deliver proteins to cells. And when they do this, they can change the function of those cells. The other thing that's really good about extracellular vesicles is that they are important in immune reactions. And so we know that these vesicles that are given off from M1 macrophages are able to convert the M2s to the other type of phenotype. And extracellular vesicles are also a very interesting new component for drug delivery. And so they have been used as a therapeutic carrier. They're in investigation as cancer therapy for infectious diseases and regenerative medicine, as well as gene therapy. And one of the things that you can do is you can load drugs into them. And the great thing about it is it takes it directly where you want it to go. So it goes directly to the cancer cells. So the problem that really has held the whole field of extracellular vesicles back is they're impractical to use as a therapeutic at this point. You can get them from in vivo culture. So basically you just grow the cells and the vesicles that they give off, you can collect them. You can get them from stem cells, you can get them from body fluids. And what you do is you collect, all these vesicles that are being put off by the cells, and then you have to isolate them, usually by ultracentrifugation. So our solution for this is we developed a cell-derived engineered vesicles, and this is actually Chris's idea from because he's the expert in membrane biophysics. But this is really a platform technology that can be used across a wide variety of applications. So what we do now is we get a blood sample. We started out by just getting expired blood products from the blood bank. And taking those bags, we would isolate the monocytes, but for the clinical trial, we're going to move to an apheresis procedure. We isolate the monocytes, we turn them into the M0, and these are kind of the neutral macrophages. Then we turn them into M1 macrophages. And then what we do, instead of isolating those extracellular vesicles, is we make the vesicles from the cells of interest by a process called nitrogen cavitation. So if you think about, if you're gonna have a soap bubble and you're gonna blow it up and it pops, what happens is all those little soap bubbles form. And that's basically what happens with nitrogen cavitation. So all kinds of little extracellular vesicles are made from the cell membranes that are present here. So then we have to do a centrifugation step to get them away from the disease or away from the debris. And then those cells are all counted and measured. And so what we think that we have done with this technology is we solved the problem. So as I was mentioning, the exosome isolation is basically you're looking for a needle in a haystack. All these different, all the different cells are putting off all different types of vesicles, and they're very, very difficult to isolate and separate out. And so they're very low yield. And again, that's what's limiting their commercial development. So what we do is our different approach is we start with the cells of interest. We do nitrogen cavitation, and we get high yields of these MEVs. And actually, our methods are 100,000 times more efficient at generating vesicles than prior approaches. So what we think is happening, so here's the osteosarcoma, here's the primary tumor, and then the most common site of metastases is to the lungs. So we have empty vesicles, and so they're empty macrophage engineered vesicles. And what they do is they find the M2 macrophages, they convert them to the M1 phenotype, and that's what results in cancer cell death. The other thing we can do with these is we can fill them with cisplatin, which is commonly used for the treatment of osteosarcoma. But any drug would basically work. They're not limited by size. So we fill them up with drugs. And then what happens is that the vesicles take them directly to the cancer cells. And so that, and the end result there is inhibition of cancer death. Okay, so now I'm going to go on and show you our data. So the first thing you always have to do with these is you have to describe them. And the two things that are important in terms of the description are the particle number and the particle diameter. So you can see the yields here are basically the same for cisplatin filled versus empty vesicles. And the diameters are also the same at about 150 nanometers. So that's where you need to have them to have those nice properties that we were talking about earlier. The size is very important. This is a picture I show pictures of my kids. This is a scanning electron microscopy of one isolated MEV. So this is, again, basically 150 nanometers. So, very exciting technology to be able to see that, and that's on the left. And the other thing we had to do is we had to show that they were getting into the cells because, nobody believes that they will work if you can't demonstrate they get into the cells. And so this is on the right, we have transmission electron microscopy. And I know it's, hard to see and you kind of have to take my word for it, but those little black dots, if you see the kind of open area in the middle with the little black dots around it, what we did is we stained the vesicles with a silver stain, and that's what's detected by the transmission electron microscopy. So you can see that they're all intracellular, they kind of localize around these vacuoles, and this is the nuclear membrane down here. So we're seeing them also going into the nuclear membranes. So pretty, at least in the field, this is considered definitive evidence that the vesicles are getting into the cells. And this is an osteosarcoma cell. Okay, so the next thing we did, I'm sorry, I have two slides for the same thing, so I'm just going to go ahead and skip this one since it's the same thing. What we did then is we did some confocal microscopy. And so what we did, so the way this is set up, these are the M2 macrophages. These are our cancer cell line. In this case, it's ovarian. And then we add the cancer cell line and the macrophages together. And so then what we do is we take the MEVs and we fill them up with dye, and the dye is the green color. So you can see these cells here. These are the macrophages. When we add the MEVs, they go inside the macrophages. And then the blue is the DAPI. So when we merge them, you can see that they're very, very quickly and really large take up into the M2 macrophages. Similarly, down in this picture here, when we look at the ovarian cancer cells, again, the green dots are the vesicles that are filled and they're being taken up by the cancer cells. And when you do the combination, when you have them both together, you have the M2 macrophages and the cancer cells, you can see really very bright staining as indicated by this green dye. So this was our proof in the culture dish that they were getting into the cells. We also did, this is a osteosarcoma xenograft mouse model. And so this is a model where it's injected intratibially. And so the redder it is, the higher the amount of vesicles in that place. So you can see that the vesicles are localizing right to the intratibial space. And when we dissect those mice, the vesicles are taken up by the tumor. They're taken up a little bit by the liver here in the blue and a little bit by the spleen, but essentially none in the lung. But, you know, again, the red spots are in the tumor, so they're indicating that they have the highest uptake. And so the reason that is, you know, the reason we went through all these localization experiments is, and the reason that this is exciting is because, you know, the big problem, well, two big problems. If you have micrometastatic disease, you can't see it on a CT scan, but our hypothesis is that these meds can actually find that micrometastatic disease and eliminate it. And the other thing they can do is if they go directly to the tumor cells, you don't get chemotherapy, you can get the chemotherapy directly to the tumor cell as well. So let's see now. So the next thing we had to do is show that they actually worked. So our whole goal here was to prove that we are able to change the M2s to the M1s. And so this is a series of experiments where they're all basically set up the same way. So M0 are kind of the parent ones. They don't, and what we're measuring here is tumor necrosis alpha. So the parents are here, the M1s are here, the M2s are next, and then this is when we add the meds to the M2s. And so basically what we want is the yellow to go up. So M1s make the most TNF, but you can see that when you add the meds to the M2s, you know, we're not converting them 100%, but we are getting significant conversion. And we basically repeated the same experiment multiple times, as you can see here. We also looked at other markers that indicate that, you know, that are markers for M2 macrophages. So again, when the green and the yellow are getting close to the same, that's proof that we're converting them from the M2 type to the M1. We also looked at cell viability. So when we do this conversion, are we able to kill some of these cells? And what we're looking for here is compared to black, we want things to go down. So in both the 10 and the 20%, we're able to show that these this cytotoxicity was observable. So this is kind of our in vitro evidence that these meds have anti-cancer activity. And again, these are the empty ones. They're not even filled with chemotherapy at this point. These are the chemotherapy filled ones. So the way that cisplatin works is it causes damage to DNA. And we measure that with this marker called gamma H2AX. And so what you can see here, and so when they're red, that's indicating that there's DNA damage, or I guess it's more pink in this situation. And so here's DNA damage caused by free cisplatin, and here's DNA damage caused by cisplatin-filled vesicles. And basically the damage is the same, but what's different here is the dose. So we use 3.7 micromolar of free and 1.5 micromolar of cisplatin-filled meds. So that's more than double the usual dose, and we're obtaining the same amount of DNA damage. The next thing we did is we went into an animal model of osteosarcoma. And so the way to think about these is, this is on the y-axis is when the numbers are getting bigger, the tumor is getting bigger. X-axis is going to be weeks of treatment. And when we look at the colors, so this is the control one. They had the biggest tumors. The red one is the free cisplatin. So at 12 weeks, they had intermediate sized tumors. The empty MEVs and the cisplatin filled MEVs had the smallest tumors at 12 weeks. So that suggests that cisplatin filled MEVs and empty MEVs are more effective than cisplatin for treating this osteosarcoma model. And, as I'm sure you all know, cisplatin still is a mainstay drug. So that would be considered one of the gold standard drugs. It's not given as a single agent. Typically, it's given in combination, but being able to beat cisplatin is in our mind, very encouraging. The other benefit of the vesicles is that they essentially have no toxicity. So one of the ways we measure toxicity in mice is we just check their weights. And again, kind of a similar graph here. Here's their weight and here's time. And what you can see is by about week 8, the free cisplatin mice started losing weight fairly precipitously. Everybody else was fine, indicating, essentially no toxicity on a very gross measure when we're just looking at weight. The other efficacy endpoint we want, the real thing that we wanted to do is identify if we could prevent the development of pulmonary metastases or lung metastases. So what we did is we did Ki-67 staining, and so we had a pathologist very much involved with this. So he picked out the regions that were known to be metastatic. And this is a very aggressive mouse model, so there's a lot of metastatic lesions here. And so what we can see here on this graph is the metastatic nodules per mouse. This is the number. And then I have it divided up by the groups. So in the control group, the mice had usually about 10. In the free cisplatin group, they had 4. In the empty MEVs group, they had even, I think, 3. And in the cisplatin filled MEVs, that actually didn't perform very well in preventing metastases, but that's so about 8. But clearly the empty MEVs were better than the control. And this was really our first go at this. And so one of the things, like when you do these experiments, if you don't have your timing perfect, like I think we let these go a little bit too long and they got too many metastases. But so I think that we also consider this promising that the empty MEVs had activity. We also looked at measures of hepatotoxicity. So these are just like regular things you would order if you go to the doctor's office, but we order them on the mice instead. So alkaline phosphatase, bilirubin, and aminotransferase. So these were all, worse in the free cisplatin. So when these numbers are going up, it's usually bad. And then this is another diagram. So looking at the histopathology. So where these black arrows are pointing at are these little red kind of inclusion areas. So these are indications of hepatotoxicity caused by cisplatin. And again, we had to get the pathologist to find them and tell us where they were. And when you look at the other mice, they really don't have these measures of toxicity, but I would say this is a fairly subtle sign. Similarly with nephrotoxicity. So nephrotoxicity is really the big problem with cisplatin. And again, we got these measures and had, when there was nephrotoxicity, it was definitely in the free cisplatin groups. But again, these little dark stained vacuoles here are actually the mitochondrial death. So again, these are indicators of nephrotoxicity caused by free cisplatin, which was not observed in the other groups. All right, and that's, more pictures of our nephrotoxicity. So with all that data behind us, we then went to, we got advice from Dr. Godali to learn how to do things the way the FDA wants them to be done so we can move into a clinical trial. And so this is a scheme of our manufacturing. What we've been doing is working on some of the manufacturing batches and getting all the conditions correct so we can run the test batches that are required. So what happens is a patient undergoes a leukapheresis. Right now, we're just purchasing those from commercial sources. Then the next step is to isolate the monocytes, and this is a positive selection with a CD14 positive beads. Then we convert them to the M0 macrophages, or kind of the parent macrophages. And then we induce them to the M1 macrophages with a couple of chemicals, interferon gamma and LPS. And then after that, we generate the MEMS. And then we have to do a bunch of testing on them to make sure that they meet our release criteria. So the release criteria that we are considering or are, based on FDA guidance is for purity, we're looking at endotoxin and different cell types, making sure that they're actually monocytes, making sure they're actually converted to the M1 type phenotype. For sterility, there's really standard things. We just do Clin Micro and also this test for fungus. And those are established and set up already in the laboratory that's manufacturing these for us. In terms of characterization, we need to count them and determine their size. We also have an identity for M1 markers, which is ICAM. And then we have to show that they work. And so that is going to be our TNF alpha readout. And so far we've made 2 batches. A lot of this is just kind of working out the conditions to make sure, because once you go to the FDA and say, this is the way we're doing it, you can't change anything. So this is mostly a lot of optimization at this point. But what we're seeing here is this is about 33 million cells. And our MEV yields from 33 million cells is like 1.2 trillion cells. And so as a pharmacologist, I'm usually thinking about micromolar and not billions and trillions. So I'm so sorry if I have to stop and think 10 times 12 is a trillion. And so, but the yields were consistent. And we're getting about 1,800 MEVs per cell. And again, as I mentioned earlier, that's about 100,000-fold improvement over current isolation techniques. We're also, our size is consistent, 164 to 154. So that's, to me, that's really good for, especially for the first 2 experiments. Our TNF release is also very consistent at 74 to 78 picograms per ml. And the ICAM that's present is 3.6 nanograms per 1 × 10⁻⁻⁻ MEVs. So again, that's also consistent. So we're really happy with the consistency in the manufacturing and just need to figure out a few more things and then do the manufacturing batches to be able to move on. So in conclusion, I hope I've convinced you that MEVs are cancer specific, and that's via interaction with ICAM, that they're able to convert M2 macrophages to the M1 anti-cancer phenotype, that they are cancer specific drug delivery, and that they're as effective as cisplatin with essentially no toxicity. And our manufacturing gets nearly complete is probably an exaggeration, but we're making progress on our manufacturing. So I'm going to go ahead and stop there and just thank a few people. So this is Namrata Anand, the one with the K on her shirt. She's in my a postdoc in my laboratory. She did a lot of this work, particularly the animal studies. Kristen Hill is next to her, and she is really working out all the details for the manufacturing. And Rob McCorkle over here on the far right, he did all the cisplatin levels for us. So, and other people in the lab also had working on different projects, but have contributed to this as well. So, so I'm gonna finish up by saying thank you. Thank you so much for the support. I really appreciate it, and promise not to waste your money. And I make good experiments and continue to make really good progress on this.
Thanks so much, Jill, for the awesome interview and super exciting. And just want to open it up now for questions. If you guys have any.
Well, could you start by just kind of explain again what the grant specifically will do to your research? Is it about, if I recall, it's about scaling up and kind of optimizing the manufacturing process to prepare for the clinical trial. Is that correct?
That's correct. So the problem, the very expensive problem we're having is when we're doing our initial batches, when we're manufacturing them, you have to transfer that manufacturing to the GMP facility, which is at the University of Kentucky. And then you have to switch to all these FDA approved reagents. Like they have special bags, and the company who makes them that is approved by the FDA, they're the only ones. They totally have the market cornered on all of this. So what we're trying, and like the LPS itself is extremely expensive. So what we've been working on is the optimization experiments to, how much LPS do we have to use? How much, how long do we have to do this for? How long do we have to be on the machine? And so we're we're finishing up those experiments by the end of June. And then with this money going forward, we're gonna run the manufacturing batches. So, and the manufacturing batches have to be done, you know, all exactly the same, all with the FDA approved reagents and in real patients, not apheresis products that we're buying. So that is the goal for this, and that is the critical step for moving us into the clinical trial.
And after you've optimized that process, do you have to do a in vivo test in a PDX model before going into humans, or is it ready for humans based on your analogies already?
So we have to ask the FDA that question. We have a meeting with them coming up in August. And I am hoping that they will let us proceed with, because one of the other kind of issues is that there's a species effect. So if we make human vesicles and give them to mice, they don't localize and they don't work. So when we do mouse experiments, we make them from mouse bone marrow. And so that is the question. So I have enough money to do this from another grant. So if the FDA requires us, we ordered these humanized mice. So those are another strange story in research. They can't be shipped until October because of the temperature conditions in New Mexico where we're getting them from. But if the FDA makes us do another experiment using a humanized mouse model, that's what we'll do. And I guess on the plus side is all these test batches that we've been making, we've been banking them, you know, storing the vesicles, they're stable for an extended period of time. So we'll be able to, you know, at least ramp that up and be able to use those.
Thanks.
Thank you.
Thanks, Jim. Other questions?
So I have another question about the use of cisplatin versus some other, I mean, I understand the MAB initial therapy and wanting to align with that, but I guess, again, I think about the people who haven't responded to MAB, and is there some, is cisplatin uniquely good for the vesicles, or is it one of many? I mean, what's the thinking behind cisplatin?
Yeah, the thinking behind cisplatin is, you know, it's still used as a standard frontline therapy for osteosarcoma. So that's why we use that. Serendipitously, we saw there's actually no liposomal version of cisplatin because it's notoriously difficult to get into a liposome. You know, so if you think about doxorubicin, there's Doxil. If you think about the taxanes, they have liposomal versions that are commercially available because they have slightly better toxicity profiles. And nobody's ever been able to do that with cisplatin. And so we were just we didn't know that. And I guess we were naive and we picked cisplatin because that is what was used. But we have apparently as discussed by according to our pharmaceutical scientists, collaborators, they think that this is a major advance in using cisplatin, even if we're not excited about cisplatin as a drug. So, but everything that we've tried to put in them has gone into them. We're doing an experiment now with CRISPR, where we put the CRISPR inside the vesicle and are able to hopefully correct the gene. So we're early on in that one. We have another collaboration with an immunologist, and they've been trying to locally deliver interleukin-2 for a very long time. So you probably know about the TIL cells that just came on the market. You know, the big problem with TIL cells is you have to get them in the ICU since they're so, you know, toxic. But if you could put the interleukin-2 inside the vesicle and deliver it specifically to the cancer cell, we could eliminate the toxicity as well. So, I mean, they're not, in terms of drugs, they're not really limited on size. We could put basically any size drug into them.
That was going to be my question. Cisplatin is, you know, can be effective, but it also has bad side effects. But this would reduce the side effects and toxicity of any drug you put in there, but especially cisplatin, wouldn't it?
Yes. Yeah, and we did a dose response study in ovarian cancer, and we're able to get, so the usual dose of cisplatin in mice is 2 milligrams per kilogram. And we were able to give them 15 milligrams per kilogram when it was inside the vesicles with essentially zero toxicity. So, I mean, that is.
Wow, that's a significant increase.
Yeah, it's a huge increase. You know, and I showed you all those, you know, toxicity profiles too. I mean, we treated probably 40 mice with osteosarcoma and at least another 40 mice with ovarian models. And we have not had a single like infusion reaction or any type of toxicity. And we've done all those, you know, electrolyte studies as well. So I am very convinced that they have, you know, essentially zero toxicity.
Related to that, are you able to somehow administer combination therapies through the MEVs, either through, I don't know if you can load more than one agent into a MEV or maybe just multiple MEVs, but different, like some MEVs have cisplatin, some MEVs have methotrexate or something like that to try to.
Yeah, so I actually have a collaborator. I was at the University of Wisconsin before I came to Kentucky, and they're very big in pharmaceutics and into developing these liposomal formulations. And one of my collaborators was famous for putting 3 drugs into a liposome. Essentially, it hasn't been commercially approved or anything, but as long as the drugs are physically compatible with each other, it's not a problem to put them in the same vesicle because. I mean, I know 150 nanometers sounds really small, but it's in terms of a drug, it's really voluminous. I guess the other thing when we do the half-life of the empty vesicles, I don't have it in my slide set, but the half-life of empty vesicles is about 150 hours in a mouse. So that's, you know, 6 or 7 days. And when we do cisplatin in the vesicles, and I'm not completely confident in the cisplatin one because we only did like 1 mouse per time point. We have to repeat it, but the half-life of cisplatin in the vesicles goes from, you know, like 3 or 4 hours to 48. So not only are you giving a higher dose, and it has more prolonged activity as well. And again, without any increase in side effects and actually no side effects.
Any other questions? You know, I actually had a clarifying question. I wasn't sure if I understood for the MEVs and developing them in because you talked about apheresis. Are the MEVs are they created, I guess, from donors? So it's not patient specific. So you know how like in NK cells, you have the allogeneic NK cells, which just come from, you just make them out of blood donors, as opposed to, you know, having like for CAR T where you actually have to manufacture out of the patient's blood product. So for this, the MEVs are just manufactured from any donor blood?
Yeah, so that's what we're going to do for the clinical study is we're going to have patients undergo an apheresis procedure. Just because the regulatory pathway is so much easier if you do apheresis and give it back to that person. Because if you do like the blood donor thing, there's a whole level of testing that probably, you know, for us, a small little group would be cost prohibitive. But I think that's where we would go, you know, if we were successful initially, that's where we would go. Ultimately, we would just use normal donors.
Because it doesn't necessarily have to be from the patient. It doesn't have anything to do with.
It doesn't, because, you know, like when we harvest the bone marrow from the mice, we don't give it back to the same mouse. You know, it's like some mice donate bone marrow and some mice get the cancer treatment. So it's not a problem. And we've never had any like reactions. We haven't measured any, and we have done repeat dosing up to, I think, 12 weeks. We haven't monitored antibodies because we haven't figured out how to do that yet. So that will be another thing we have to figure out. But again, we don't have any like infusion reactions or loss of activity.
So it's really more for just kind of the regulatory
Yeah.
expedite to kind of do it, the apheresis for each patient. but
Yeah.
really you can do it. Okay, got it.
Yeah, definitely a regulatory strategy at this point.
Jill, additional clarification, the shelf life of the vesicles, what would that be once you go start to finish? What do you expect?
Yeah, well, what we know so far is that they're stable at 4 degrees for at least 3 months without losing any activity. If you
Wow,
freeze,
interesting.
if you freeze them, it's not as good. And the other, like, I don't have all the data put together for this, but the kind of hot off the presses data is that they're lyophilizable. So the empty MEVs for sure, you lyophilize them and they are just as good at, we use TNF production. And they are just as good as the ones that you didn't lyophilize. I'm not 100% sure about the cisplatin yet. We have to do measure and see how much cisplatin we're losing. But like our preliminary things show that if you lyophilize the cisplatin filled ones, they also retain their activity. But we need some more data points on that. So that's not a for sure, but for sure the empty vesicles can be lyophilized.
Thank you.
Any other questions?
Just one other thing I was gonna say, going back to the last question. The other thing that we can do, what we're planning in the clinical trial is to give 6 doses. And so we can actually take the monocytes and freeze those after we isolate them. And because the blood bank does that all the time, so it's very standard. And so then we can just thaw out when we want to do a dose, we can just thaw out the number of monocytes that we need and make the vesicles from that dose. And we think 1 apheresis is going to be enough for 6 doses. Yeah, I'll jabber on about this all day, so thank you.
It's fascinating because it's a very new, area. I've never heard of MEVs before until reading about your proposal. So it's really interesting and so many advantages.
Yeah, we are
Has
really
this,
excited.
Jill, has this been used previously by anyone else that you're aware of?
So
The
there is
MEV.
not MEVs. There has been one clinical trial for COVID that used the other vesicles that are given off. I can't remember what immune cell is giving them off, but the endogenous vesicles that are given off by some immune cells were collected and then administered in a COVID study, and it was effective. But so far, that's the only vesicle study that's made it into clinical trials. And they definitely didn't use our method of making them from the cells. They isolated them from huge. So if you think about it, it's like a vat the size of this room is a dose for a patient for that procedure, for that method of isolating vesicles.
Thank you.
Okay. Any other questions? And if you think of them later, you can always, I'm sure, email them. And Dr. Kolesar will be at Factor, so if you're going to be there, you can ask her in person and chat with her in person, which will be lovely. And if you didn't get enough, we did record it. I started it a little late, but I did press record at some point before you got started. So we'll send out a link to the recording in the Family Fun Flash that's going out tomorrow. You can watch it on your weekend, Friday night movie night.
Thank you. This was really
Thank you.
great. You took a confusing bunch of papers and made it very, very understandable. So thank you.
Well, I just want to
It's
thank
exciting.
you for being here, and I appreciate your support. It really means a lot to us because it's encouraging to us. When you work in the lab all the time, you think it's great, and yeah, so it really means a lot to us that you thought it was worth funding. So thank you very much.